This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, or TWiV, because there's really no syllables in TWiV. This Week in Virology, episode 1221, recorded on May 23, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here, and it is cloudy and rainy here, as it has been for the past couple of days, which was good. We needed some rain for springtime, but it's supposed to clear up. It's 52 Fahrenheit, 11 C.
15 C and cloudy here in Chelsea. Also joining us from Montreal, Canada, Angela Mingarelli.
Hello, everyone. It is also cloudy and kind of cold. It is 48 F and 9 degrees Celsius. There was like a glimpse of what could be summer last week, and then suddenly it got freezing again. I turned off all the heaters in my house, and then 2 days ago, it was freezing again. I had to turn them back on. But next week it's supposed to be nice and warm again, so we're looking forward to that.
We've been flipping back and forth between heat and AC here, too.
So by July, it'll be very hot in Montreal,
Yes.
right?
Yes, for ASV.
Yeah. Very hot. The convention center is air conditioned, so it doesn't
Definitely.
matter. And that's it. All three of us. One more than last week, you know. You know, if you enjoy these programs, these science programs, where you get science fact through our eyes, right? But still science fact. We don't do misinformation. We don't do lies. We'd love your support to continue them. We have grown, of course. We have 9 podcasts, I think. And so the expenses have grown as well. And we'd love to have your support. Microbe.tv/contribute. ASV 2025, speaking of ASV, hosted by McGill University. And they were smart enough to do it at the Palais des Congrès de Montréal, because doing it on campus is just crazy. You have to be running all over the place. Do you know the organizers, Angela?
I actually don't know. What are their names?
I don't know.
Do you know their names offhand? No, right now, no.
Offhand,
I know a lot of
don't.
people are going. Everyone I talk to is going, but
might as well go. It's there,
It's
right?
there. Yeah.
Exactly.
So the website, so the dates are July 14th through the 17th. I've got my hotel. I got my plane. I have to leave from Newark. I hope the Newark airport is still running then in July. It's such a mess. Alan, 4 controllers out of 12.
Yeah.
Ridiculous.
Yeah, and radio systems going down. And
that 90-second blackout seemed
yeah.
terrifying. That many planes coming and going. And that
their
systems are so rudimentary. Still.
Yeah, I mean, there are multiple layers of protocols in effect, so they're not all going to fall out of the sky or run into each other just because the radios went down. But they can't they can't make approaches to the airport if the radios are down because they can't sequence the traffic. So then they're just going to be kind of hanging out.
Anyway, hopefully by July it'll be sorted.
Yeah.
It's July 14th through 17th. The website is asv.org/asv2025. Check out the program on the website. You can see special events and workshops. In addition to there's morning plenary lectures, and then in the afternoon there are all kinds of overlapping sessions. And the special events included TWiV, of course.
You could, I think Amtrak goes again to Montreal, right?
I could always hop on an Amtrak. That's right.
That's a beautiful train ride right up the Hudson.
Yep. And you don't have to wait at the border, I guess, right?
You do have to wait. Well, it was many years ago that I did that. I biked up to Montreal from New York on a vacation from graduate school. Took me a week.
Biked?
I biked.
That's amazing.
It was fun. I camped out in campgrounds and just biked up because I couldn't afford to really go anywhere on vacation. I was a graduate student. But I thought, I really like long bike rides. I'm going to go bike up the Hudson Valley. It was gorgeous and did that. And then I took the train on the way back. And the train coming back stops at the border and customs goes through and, checks everybody in.
The standard registration ends June 30th. After that, the rates go up. The reservation deadline for the Campus One MTL residence hall has been extended to June 12th. Sign up by adding the reservation to your meeting registration. So you can stay on campus if you want. I've never done that. I always stay at a hotel because I'm a snob. I found one that's 10 minutes walking from the Palais des Congrès.
It's perfect.
Because that's good. 10 minutes is good.
I'll come pick you up every day, Vincent.
You don't. If you're gonna walk, yeah. Don't drive, right?
I take the metro. I take the subway. It's way faster in Montreal. Driving is crazy trying to find parking downtown. Unless you're in the Palais des Congrès, you could just pay for parking. But the metro goes right there, actually, into the
come by and
center.
We'll walk to the Palais together. Sure,
Mm-hmm.
that'd be cool. Couple of news items. Man,
yeah.
I have the first one is a couple of New York Times articles, so I have them open here on my laptop because I have a password there, which was given to me by one of my Columbia students. So Columbia gets free access to the New York Times for your life.
Oh,
Wow.
nice.
Which is pretty good. And one of my students gave me because they said they have an extra one. Probably shouldn't tell anyone that, so get in trouble. Anyway, first article of the Times. Trump has cut science funding to its lowest level in decades. And this article concerns the National Science Foundation, which supports a lot of fundamental research. You know, all the human health, medical related stuff NIH usually does. But if you want to work on fruit flies or worms, NSF is the place to go. Fundamental research.
And outside the biological sciences, too.
That's right. They have a lot of different areas. And so through May 21st, the number of grants funded are down 51%. And they touch every area of science. So they include education, computer science, math, physics, chemistry, biology, social sciences, geosciences, technology, and engineering. And they have a nice breakdown of all the cuts here across the board. But for example, the biggest canceled funding from in-progress grants, the biggest drop is in STEM education, which is the dumbest thing you could ever do, right? You need to have people educated, and it's down $650 million. And then math, physics, and chemistry are down substantially in new grants. So really bad. This is just NSF, of course. We've talked about NIH before.
Mm-hmm.
It's a form of bigotry, actually, to cancel certain kind of grants and not others.
Yeah.
Right? But this administration doesn't care. Biology is down 52%. It's a great article with lots of nice graphs, but you may not be able to access it. All right, someone put in the next one. Go ahead.
Yeah, I also added, on that same line, Harvard researchers. So this is in Nature, an article in Nature that came out, a news article today. Yeah, today's the 23rd. Basically just highlighting over 1,000 grants that the Harvard University has lost, with an amount summing to over $2.4 billion as well towards their research funds. And this is just obviously devastating. I mean, Harvard is one of the wealthiest universities, obviously, but this still is very significant. Even though they have their endowment, they still depend on a lot of those grants for research funding. And then I also added another one, kind of linking the federal judge Alison Burroughs talking about so this is a CNN article on the Trump's administration ban on Harvard's ability to bring in more international students, which is crazy.
And they cancel everyone's visa, right?
Yeah, well, I'm not exactly sure how they're doing
trying
it, but
to
I think it's
block
enrollment
Harvard from getting student visas.
Yeah,
exactly, and bringing in more international students for
Which is illegal.
For the following year. Exactly,
It's completely illegal.
Well, so is the cutting of grants illegal. It's going to take forever to get through the courts, right?
Yeah,
and then the courts will hand down the decision, and they'll just ignore it because the Department of Justice is run by a lackey, right?
Right, but most of what's being done is either illegal or certainly challengeable.
I mean, this Harvard thing is just because they won't hand over the reins, so they're doing all this punitive stuff, which is the most childish way to run a government. And if you Americans who voted for him want that, it's ridiculous. go live somewhere else.
Yeah.
But I heard that students who were here on visa, they were canceling them and deporting them. Right? It's
I heard
just,
that as well, but I don't have that information firsthand. It was
yeah.
more just anecdotal, but it's pretty bad.
Then I
Even
have a
for-
Nature article, a news article. NIH killed grants on orders from Elon Musk's DOGE. So and this is sworn testimonies from NIH officials that the DOGE representative directed them to terminate specific grants. The DOGE official, which is like, doesn't know anything.
That's an oxymoron, by the way, DOGE official. It's not an actual department of government.
Right.
It's completely made up. It's just Elon Musk, as Trump has let Elon Musk get access to all these departments in a completely unconstitutional, non-regulatory setting. None of this is being done in any legal manner.
What really infuriates me is the rationale. There's a spokesperson in the White House. Paying so-called experts to deliberate bad ideas for hundreds of hours is exactly the kind of waste that DOGE is eliminating. This is Kush Desai. You are such an idiot. You have no idea what goes on with a grant. Just, it obviously that's just a stupid excuse. And many people will think, oh, that's good to cut those people sitting around with bad. No, that's not what science is. Disgusting. Yeah, but this, and then they said to the head of NIH, Bhattacharya, they asked him about the cuts. I don't personally review grant lines and say I cancel this grant. He said there's a process. There's no process. There's DOGE just cutting stuff in certain areas that they don't like. This is just insidious and horrible.
It almost seems like a nightmare that can't be true, that we must have to wake up one of these days and that it was all some terrible dream.
It's just really annoying. It's very upsetting to me to wake up and think about this and think about everyone's lab is just being terminated. Many people's lab is being terminated, and it's just not justifiable because many of them are very productive, right? And we're going to hear today about 2 wonderful stories done in laboratories, probably supported by NIH funding. And, I wish you would not lie about the reasons for canceling something.
My pick touches on this.
But the interesting is we have a paper on SARS coronaviruses, and they say we need more research to improve our response to COVID and other upcoming pandemics. And they've canceled all the COVID grants, which were not just about COVID, but also trying to predict and get ready for the next thing. And they canceled them.
Yep.
They're idiots. I know I rant about this every week, but have to, because I'm hoping that somebody new listens who doesn't know this, and it will change their vote.
Hopefully, because right now there are so many viruses out there. It's not maybe, they're definitely out there circulating in animals, wildlife, wherever they are. We have no idea. They could potentially spill over into humans, and we don't even have money, or nobody cares to sample them. But then when it happens again, it'll be our fault. Even though we didn't have money it'll all be our fault, which is the most hilarious part.
And by the way, it's not so much getting people to change their vote as getting people to actually vote.
That's true.
Also, yeah.
I don't know how likely that is. How much does the voting vary from, election to election? Does it vary?
We'll talk about that on my pick of the week.
Your pick of the week? Okay.
Yes.
Very good. Okay. It says the world is potentially a wonderful place. Humanity has done amazing things, but on a small level, when you drill down, man, do things get screwed up. Just people don't realize how unique humanity is, and they shouldn't be messing with it in all the ways that they do. Anyway, into science. We have a Journal of Virology paper now. This is in the last couple of weeks, we've done 2 Journal of Virology papers, which is good because this is where the nuts and bolts happens.
Yeah. Join the JV team.
Maybe not as splashy as other journals, but nuts and bolts that are important and will allow us to educate you about the background. I think that's the best part that I like about these papers. It says endemic coronavirus infection is associated with SARS-CoV-2, FC, it's not a curse word, FC receptor binding antibodies. There's no asterisks in the FC.
No.
David Bean, Yan Mei Lang, Frida Avila, Xiaobao Hu, Arcana Asundi, and Manish Sagar. And then they put the bloody...
All from Boston University.
Thank you. Page 15.
Yes. At least on this paper, at least in JV, they give you a link.
Oh, there is
author
a link
affiliations on page 15, and you can click the link if you're reading the PDF. In a couple of the Nature journals now, they just say author affiliations appear at the end of the paper.
Yeah, I don't know why.
And
they
you
started
can't
doing
just
that.
go to the end of the PDF because that's after all the supplementary figures, and the
Yeah.
authors were 20 pages before that. You've got to scroll through to figure out where these people are from.
Or maybe we're the only podcast in the world that does
Reads the
papers,
author affiliations,
right?
Yeah.
So who cares about us?
Which is
All right,
crazy because
so-
it's like the most important part is to know who did the work.
Yeah,
Who did the
and
work?
Especially
Who-
the poor graduate students in many cases that have
Yes,
dedicated
right.
their
You got to acknowledge
years,
the people.
exactly, years of their life to this.
Yeah.
In some cases,
Well, I also like to know where it is and
Hmm.
to illustrate the collaborations between multiple institutions, right? I think it's great.
Mhm.
So as we know, we've talked about before even SARS-CoV-2 emerged, we knew there were multiple endemic human coronaviruses. They're circulating. Well, SARS-CoV-2 is already endemic, but these endemic coronaviruses caused pretty mild respiratory disease, and they have names like 229E, HKU1, NL63, and OC43,
Because they were all named separately.
all named
Mhm.
separately.
Mhm.
And
We couldn't
OC
do like the hepatitis researchers did. Was
Yeah,
just
right.
letter them.
So there's evidence suggesting that people get infected every 18 months to 2 years or even more frequently, as you'll see. And it was suggested that the immune responses against these endemic coronaviruses are cross-reactive with other coronaviruses. And people have found that if you had a previous endemic coronavirus infection, you had less severe COVID disease. This is before we started vaccinating people,
Mhm.
and that kind of mushed that up. But
Vincent,
we don't
can we-
understand why there would be cross protection.
Can we highlight? I know we've said this in the past before, but just for the listeners again, that common cold. So, what viruses comprise the common cold? Because these endemic coronaviruses do comprise the common
Yeah.
cold. When people say common cold, it's actually the 4 viruses you just mentioned, but it also includes RSV, parapneumoviruses, adenoviruses, enteroviruses, rhinoviruses, the
yeah, and other-
influenza itself also could be in there,
Sure,
although people
sure.
call it flu. But so just importantly that these are viruses that are circulating
So-
and they form part of the common cold.
A common cold is basically an
Common
upper respiratory tract infection, right?
Yeah.
Mhm.
Upper
Mhm.
tract for nasopharynx.
Mhm.
And it's annoying because you get sore throat and cough, and your nose drips. You lie back in bed at night, and it drips back in your throat, and it tickles and wakes you up, makes you cough. It's very annoying, and a lot of people
Your
stay
nose
home.
gets all red and itchy. You're blowing your nose. All of these
Yes.
things. It's not one
Makes you
virus.
snore, and then your spouse
Could be
elbows
many.
you. Yeah.
Exactly.
So it's mild disease, and they say some very interesting things about it. So because it's disease, nobody goes for medical care, and so
Because it's mild, yeah.
so mild, and so yeah, I'm okay. I don't need to get medical care anyway. If you did, there would be nothing much to do, and so you're not widely testing for endemic coronaviruses, right? Like you are for influenza during the flu season, and so actually knowing who's infected when is not so easy. But they use their approach here is they combine PCR documented endemic coronavirus infections with looking for antibody against the endemic coronavirus nucleocapsid to say who has had a recent endemic coronavirus infection. And then they look at cross-reactive immune responses to SARS-CoV-2 antigens from people with or without a presumed endemic coronavirus. ECOV, they call it. ECOV. It's like electronic device.
And the point of doing all this is we want to know what the the correlates of immunity are for just coronavirus resistance. Because
Right.
what we'd love to have is a vaccine you could give people that would be good against a whole wide range of coronaviruses. And this tantalizing clue that people who had a common cold coronavirus may have had milder cases of COVID suggests a route to get to that.
Right.
So
they
one
test,
of my
yeah,
Sorry, one of my questions, they say nucleocapsid of ECOV, but ECOV is 4 viruses.
yes.
So is it that they pooled the data as for nucleocapsid of each, or is it the same in each? Because they didn't define that.
It's a good
I
question.
don't know, but I think they're very, very similar, right?
Okay.
So you could use one and they probably cross-react because, antibodies against SARS-CoV-1 nucleocapsid will light up SARS-CoV-2 nucleocapsid
Mhm.
infected cells. They're so conserved, right? So one is probably
Okay.
enough, but I don't know. Yeah, I don't know what they did.
And I think one of the things that kind of powered this study is the availability of these tests that you can do that are like a whole respiratory virus test that does kind of a panel of common cold viruses and flu viruses and pretty much everything Angela just listed. So then that is being done in the clinic, and that gives access to the kind of data they needed to pull these patients out.
Well, actually, they say that these multiplex panels aren't used much.
They're not,
right?
yeah.
In a hospital setting, they would be,
Yes.
but not you go to your doc because they're $800 a pop.
Yes.
And it's only in a hospital setting if you have a very sick person, you need to know what's infecting them. So that's not useful for this, but they have done their own screening as we'll
right.
see.
What I meant is that they kind of based this on that.
Yeah. So the primary goal is to compare immune responses, SARS-CoV-2, among individuals based on whether they do or do not have ECOV infection. So they collect samples from people with previous SARS-CoV-2 exposure, right, or prior COVID vaccination but no documented SARS-CoV-2 infection, or no prior COVID-2 antigen history whatsoever. And then they want among those to identify whether any people have had undocumented infections that's going to screw up their study because they're going to get rid of those. And so they want to identify these people who have had undocumented asymptomatic infection. So they measure plasma IgG against the SARS-CoV-2 receptor binding domain and the SARS-CoV-2 nucleocapsid by ELISA. So the receptor binding domain is you make a lot of antibodies to it, and it's the more variable part of the spike, as we will see.
And that's the focus of most of the vaccines, right?
Right. The whole spike is in the vaccines, right?
Right.
But most of the antibodies you make are RBD.
Or RBD.
Not all, but many. So then they use this data to set cutoffs for the IgG levels that could differentiate and detect people with undocumented infection. And most of the individuals with either a previous infection or vaccination had elevated antibodies. And all the pre-pandemic samples, right, where they can't be infected, pre-pandemic, were below the set cutoff that they made for exposure. So they use those numbers then, and they have 7 individuals who were originally in the no SARS-CoV-2 group, which turned out they had a previous SARS-CoV-2 infection. So they were excluded. You can leave now. You can go home. And then another 6 were in people that this was people with prior COVID vaccination who said they weren't infected. They turned out to be infected because they had elevated nucleocapsid levels, right? Because you wouldn't have elevated nucleocapsid antibodies if you're just vaccinated. So they got rid of those too. They ended up with 94 individuals who were not previously infected with SARS-CoV-2. Okay. And then they have other controls as well. So what they want to do is look at anti-SARS-CoV-2 immune responses among these people with and without ECOV exposure in the absence of SARS-CoV-2 infection, because that screwed the whole thing up if you had SARS-CoV-2. You just want ECOV and nothing else, right? So how do you identify people with a recent E. cov infection? So they say that these infections probably occur every 1 to 3 years. And the antibodies decline slowly, and then you get reinfected as a consequence, and you get mild disease. So they took this time frame and looked in the Boston Medical Center electronic medical records and identified 22 people who had documented E. cov RNA that's right, Alan, you're absolutely on a prior multiplex PCR test, right? Less than 550 days before the sample collected for this study. These were classified as having recent E. cov infection. So it's really important to have a electronic medical record system to be able to do these kinds of studies, right? And then to find some other people with recent E. covs and not SARS-CoV-2, they measured plasma IgG against all 4 of the E. cov nucleocapsids. So there you go, Angela. They did all 4.
Okay, they did. I missed that then.
Independently. And the top 25 individuals with the highest nucleocapsid antibody levels were classified as having a recent E. cov, but not SARS-CoV-2. So in other words, you had one set based on multiplex PCR and then another set based on antibodies to nucleocapsids. Okay. By the way, Alan and I were at BU many years ago doing a documentary. Remember,
Yes.
Alan?
Yeah.
The needle. We went to the needle.
Yes,
we
did.
threaded the needle
before it was open, before it was doing actual lab work in it.
Yeah, good. It's a good documentary. You should check it out. Okay, so now they do the experiments. They look at cross-reactive immunity after a recent E. cov infection. And they say, we're looking at antibodies because these are what we know is a correlative protection against SARS-CoV-2 and severe COVID. No difference in anti-SARS-CoV-2 RBD antibodies and SARS-CoV-2 S2. So the spike is divided into 2 parts, S1 and S2. And or IgG antibodies in people with and without a recent E. cov infection. Okay, so no difference in antibodies that hit the SARS-CoV-2 RBD or S2 in people with and without an E. cov.
Then S1 is the RBD.
Basically, yeah.
And S2 is a more internal part that's less exposed and you generate fewer antibodies against, typically.
And they also did neutralization experiments. So they have, they actually use a VSV with the SARS-CoV-2 spike. So they don't have to do this under higher containment. And there's no difference in neutralization titers between people with or without a presumed E. cov infection. So this is actually contrary to what other people say. Some people say if you've had an E. cov infection, you have neutralizing antibodies against SARS-CoV-2, and others do not. So it really depends on the cohort you're studying and your reagents. But here they don't find any difference in neutralization. So apparently E. cov doesn't help you with neutralizing antibodies against SARS-CoV-2.
But neutralizing antibodies are not the only useful kind.
So what else is there, Alan, besides
Well, I mean, a neutralizing
antibody
stops the virus from infecting anything. But you could still have an antibody that sticks to the virus and just hangs on there. And the immune system has mechanisms for recognizing antibodies and saying anything tagged with an antibody has got to go.
And of course, there's also T cells, right?
Yes.
That could be involved. And that's what they look at all of these things in this paper. So we've ruled out neutralization. And then they say, right, the antibody is a Y-shaped molecule. And the Y part, or the 2 tops that make up the Y, or where the antibody binds to an antigen. And the bottom part, which is straight, right? A Y has a straight bottom. That's called the FC portion. And there's receptors for that on various cells in our bodies. And when an antibody, say, binds a virus and then the FC engages a receptor on some kind of cell, that can be a form of clearing of viral infection. So, for example, antibody-dependent cellular cytotoxicity. There's certain cells where if this complex is binding to them, so you have an FC receptor on a cell, and that can activate those cells to be cytotoxic.
But isn't it the Fab portion that binds to
Yeah.
the actual antigen in itself, and then it's
That's right.
whatever the myeloid cell, let's say macrophage or anything that's going to mediate this death, ADCC, which we haven't explained yet, but that will then bind to the FC portion, right?
Yes.
Because it's like the top part of the Y versus the bottom part of the Y.
And Angela, do you know why they're called FAB and FC?
Oh, I did. Well, I know that the C stands for crystallize, doesn't it?
Crystallizable.
Exactly, and FAB.
But I don't remember the answer actually, but I think it's because there's an enzyme that cuts between the two. And in the early days when they were trying to look at the structures, they found this. And I think it might be bromelain, but I'm not sure. But it could be something else.
I think FAB is the antigen binding fraction.
Antigen binding
fraction or fragment.
Right.
And then
FC. But in the old days, before recombinant DNA, they had to take protein and cut it with a protease. And that was it.
So you can think of the antibody as an adapter.
Basically.
It has the variable region that binds to whatever that particular antibody binds to, but then it has this constant region, this FC tail, that's going to be the general purpose flag for, hey, immune system, this has got to go.
Mm-hmm.
So they decided to look at this, if any of these antibodies, which don't neutralize infection, remember, antibodies that are present in people with E-cov infection, whether they can do this FC-mediated activities.
Mm-hmm.
So they use an ELISA method to measure SARS-CoV-2 specific antibodies that bind a specific FC receptor. There are a couple of them. And the one they do here is another alphabet soup, FC gamma R3A.
Which, depending on the font, can look a little like F gorilla.
It can, yes. Or Cyrilla. Interesting. And so they say they picked that one because it's a predictor of NK cell-mediated antibody effector function. So I think what happens, Angela, and I've totally forgotten, but the antibody binds the NK FC receptor, and then the NK cell is activated to kill infected cells.
Exactly, cross-linking on the NK cell, and then
Something
it's
like
activated via that portion you just said. It's also called CD16, which is way easier to remember.
Yes.
And it's all over lots of myeloid cells like NK cells, macrophages, and neutrophils. So it activates them, and then in some cases, they release cytotoxic granules like perforins or different things depending on the cell type, which will then kill the cell that is it is attached to.
Yes, so the antibody need not just bind a virus particle. It could bind an infected cell with the glycoprotein on the surface and then kill it that way, right?
Mm-hmm.
So they look for these antibodies, CD16A binding antibodies, okay? So people with prior SARS-CoV-2 spike exposure, so infection or vaccination, they have higher levels of anti-SARS-CoV-2 RBD and S2 antibodies that bind to FC gamma R3A. All right. So SARS-CoV-2 infection or vaccination leads to an elevation of these FC binding antibodies. And then these elevated responses are not different between people with either an infection or vaccination, SARS-CoV-2 infection or vaccination. People with or without a recent E-cov infection had similar anti-SARS-CoV-2 RBD, FC gamma binding antibodies, but the anti-SARS-CoV-2 S2 FC gamma R3A binding antibodies were 8-fold higher, and the ratio of antibodies to IgG was 85-fold higher in people who have E-cov compared to those who don't.
And this is so we're now, kind of the money shot here is people who had E-cov but haven't had SARS-CoV-2,
right.
have the 8-fold higher level of these anti-S2, right, which is not the usual response you would get against the spike receptor binding domain if you were vaccinated. They have elevated levels of these anti-S2 antibodies that can bind FC. They're not neutralizing antibodies,
Right.
but they have this adapter function.
And these are reacting with SARS-CoV-2 S2,
Yes.
as well as binding the FC receptor. And they were found in people who have no prior SARS-CoV-2 infection or vaccination, only ECOV infection. So that's the key. You have an ECOV infection, you make antibodies that can react with SARS-CoV-2 S2.
So these are cross-reactive antibodies that you generated in response to your common cold coronavirus, and now they react to this particular part of they're not neutralizing, but they still have an effect.
So that's basically the story. They're not
Mm-hmm.
neutralizing, but they are binding to these FC receptors. Okay. Then they want to know whether these so these antibodies are originally directed against the ECOV spike, right? Because that's people get infected with an ECOV and they make antibodies against that spike and they cross-react with SARS-CoV-2 S2. And they want to know if these binding antibodies account for the anti-SARS-CoV-2 S2 Fc gamma antibodies, right? Because well, we know that they're there right now. So they look at they have these antigen-specific ELISAs to measure antibody levels against the ECOV spikes to see the antibody response against those, right? Because we don't know what that is so far. So they found IgG levels are higher against the 229E spike and the OC43 spike in individuals with a presumed recent ECOV infection. So that's good. I mean, in fact, that's how they were recruited. Some of them were recruited into the study anyway, right? But now they're doing it all at once. And for 229E, the spike specific Fc gamma binding antibodies were also higher in people with a presumed ECOV infection. So they get a statistical relationship showing that these Fc binding antibodies are against the spike. And of course, there's the ones that can mediate binding to SARS-CoV-2 spike.
And the S2 region, if I'm not mistaken, is more conserved among coronaviruses than the S1 receptor binding domain, right?
That's correct, yeah.
Which would explain probably why these are
Yeah.
cross-reactive, because they're binding to a more constant region.
Yeah, the RBDs are probably very different, and
Yeah.
you're not going to get cross-reactivity because, I mean, SARS-CoV-2 variants that arise, the antibodies are against the RBD, and they don't cross-react anymore in some cases. So then they look at the reactivity of these antibodies against the spike to see how much cross-reactivity they have. No correlations in SARS-CoV-2 and ECOV Fc binding antibody responses in individuals without a presumed recent ECOV infection. So that's what they like to see, and they do statistical tests to measure that. They also measure ECOV Fc binding antibodies after depleting SARS-CoV-2 S1 or S2 binding antibodies. Okay, so you have a mixture of well, you want to know if these binding antibodies are the ones that are going to confer the effector function. So they deplete them from the serum samples that they have, and depletion with SARS. So they deplete with the antigen, with the S2 or S1 attached to beads. So they throw those in the serum, and then they let them bind, and the beads can be centrifuged out, and it will pull out the specific antibodies against S2 or S1. And so depletion with SARS-CoV-2 S2, but not S1, significantly decreased the 229E and OC43 spike Fc binding antibodies. All right, so you're pulling out the antibodies with SARS-CoV-2, and then you look for and the reactivity with 229E and OC43 spike, and it's gone. So because they're cross-reactive, right?
Yeah.
They're binding originally to the OC43, and they're also binding to to SARS-CoV-2, and S2 pulls it out again, consistent with what we've already said, because these cross-react with S2.
Yeah, so this is a confirmation of what we suspected was the case, that the S2 binding antibodies are the key.
Mm-hmm.
Right. So they conclude that, or they suggest that these ECOV antibodies that are able to bind FC gamma receptors go up after an ECOV infection, and they correlate with increased cross-reactivity against the SARS-CoV-2 S2. In fact, they looked at levels in this analysis, which I didn't go into statistically, and that's how they can make that conclusion.
Do we know how long it was from infection, these people with the ECOVs? Because this was in a bank where they had the information about the multiplex, right? So I wonder if it was, obviously it wasn't all the same timing. I'm sure these people, it was quite diverse, but I wonder how long in time they would be protected, potentially, at least with these antibodies also for cross-reactivity,
so they said
SARS-CoV-2.
They had a positive multiplex test less than 550 days before sample collection. So that's the
So it's like a year and a half. Yeah.
Okay.
And that's because the antibodies go down in a year and a half, then you get reinfected. So you don't want that to complicate it, I suppose, right? All right. So then they say, what about T cells? So ECOV infection induces cross-reacting antibodies that bind SARS-CoV-2 S2, and these can bind FC receptors. So can ECOV induce T cells that react with SARS-CoV-2 T cell epitopes, right? And they have shown that, and others have shown that T cell responses induced by SARS-CoV-2 are associated with lower ECOV infections. So the opposite of what we've been talking about so far. The people, if you got COVID, you have a lower chance of getting ECOV. I think it's just disease, not infection, because it's not likely to be sterilizing. But anyway, so they take out, so they have to have blood available. In fact, PBMCs from these patients. So not everybody has that. And then they take these PBMCs and add antigens and see if they respond to them. And so if you've had a previous exposure to SARS-CoV-2, again, either infection or vaccination, you have higher CD4 and CD8 responses to the SARS-CoV-2 spike and nucleocapsid, because we know there are epitopes, T cell epitopes in both. But there's no difference in CD4 and CD8 levels against SARS-CoV-2 spike or nucleocapsid, with or without presumed ECOV infection. So they're both the same. So there's the cross-reactive T cells are not boosted after a recent ECOV infection. It's just SARS-CoV-2 infection that does that. So antibody neutralization is ruled out. T cells are ruled out. Looks like antibodies that bind FC receptors. Now, the authors admit that they have not done a functional assay to prove that. Right? That's what makes this a JV paper as opposed to Cell. And I don't mean to be derogatory, but it's a hard experiment to do. I don't know how you would. I guess you'd have to do it in vitro, but then you'd need cells from these patients, and maybe you don't have them or whatever.
And it's getting harder and harder to find patients with no SARS-CoV-2 exposure.
Yes.
Yeah.
Yes, that's right.
True.
Novids.
People
who say they're novids, I always say, well, how do you know, actually? Because because here you saw some novids who are actually IgG positive. And, right. But anyway, so here we have an interesting observation that it looks like antibodies, cross-reactive antibodies induced by ECOV infection can bind the S2 part of the spike of SARS-CoV-2, and they can also bind FC receptors. So that could, in theory, mediate these cellular mechanisms that would eliminate infected cells.
And this points toward a possible strategy for if not a pan-coronavirus, a multi-coronavirus vaccine, where you might try to use something like an S2 based or a consensus S2 based antigen, stimulate immunity against that, and instead of doing tests where you're looking for neutralizing antibodies against all the coronaviruses, look for FC binding antibodies against as broad a panel of coronaviruses as possible, on the thinking that that should be or may be partially protective. And if we've got a novel pandemic breaking out, partially protective is a whole lot better than not protective at all.
True, because it'd have to be partially, because T cells, even on this case, obviously, I don't think it's surprising that T cells weren't different because the epitopes are so specific in like each clone, each T cell. But if you don't have a T cell response, even in like a pan coronavirus vaccine, then it would definitely wouldn't be fully protective. But like you said, if you have some antibodies at least already hanging out in the blood, already ready, then that would definitely, potentially improve disease outcome.
And if you can develop something like that, kind of on speculation in advance, and then have a stockpile of it when the next coronavirus breaks out, which is going to happen at some point, then you can distribute that initially while working on a more specific vaccine that would be more protective. Probably save a lot of lives if that works out.
True.
Now, there's an interesting twist to this, which I want to mention. So the binding of FC to FC receptors depends on fucosylation of the FC domain. So addition of fucose, the sugar, right, that improves the binding. And so they say, in which I didn't know, that as we understood getting into the COVID pandemic, as we understood that FC mediated mechanisms were probably important, the therapeutic monoclonals that were made and used for a while all were made with fucosylated FC regions, so they're more capable of mediating these activities, right? So is it fucosylation or afucosylation that makes good binding?
I think it's afucosylation if it's not fucosylated.
Okay, so if there's no fucose there, so these FCs can have fucose, but it binds better without fucose. So the monoclonals were made without fucose. So they would have this. We didn't know the contribution, right, because it hasn't been tested, but we had an idea that it might be, and so they did afucos just in case, right?
So it's best to get the fuke out. Yeah.
We only measured levels and not the function of FC binding antibodies, and thus we can only speculate about increases in ADCC and ADCP. Okay, which is fair, but it's still an important observation.
Yeah.
Right? So that's why I thought this was interesting. And, we don't talk much about endemic coronaviruses, ECOVs. But,
I mean, they're also kind of hard to do in mice. I speak from experience.
Yeah, you tried?
NL63, I tried to mouse adapt for like a year. It didn't work. It grows so poorly in culture. Like, it's almost impossible. And you even order it from like we ordered the virus multiple times, but the titer that they have is so low that you freeze thaw once, you lose half your virus, and then it won't infect anything. It's terrible.
Nice.
So even they can't grow it well at, what is it, BEI Resources, I think. It's part of the NIH.
Yes.
And
CDC.
229E grows way better. 229E is, but much better. And OC43, but NL63, and then the funniest part is that I think I told this at TWiV 1000.
Yes.
We did virome sequencing and I had the virus. I literally, because we were doing like another experiment on like nasal respiratory viruses, and I actually had NL63, and I was like, oh, I could have just like spit in a dish on myself. I'm joking. But it's in mice. It's not that easy. At least some of the endemic coronaviruses. So I was thinking if they could somehow do functional experiments like in mice. But some of the viruses is not so easy. Yeah, they're not mouse adapted.
So you're done with that project?
For now, yeah. Now we're doing T cells.
Okay.
Other T cell things.
With ECOVs?
No, not ECOVs. No, LCMV normally.
I see.
And like flu, influenza, mostly.
Okay. Now we have a paper in the Journal of Experimental Medicine from 2019,
Both of
And,
the papers today are open access, by the way.
because I thought this was a very interesting finding that, and it doesn't matter how old something is. If we haven't talked about it, it's fine, right?
Yeah.
So this is a very interesting observation. The title is Inherited IFNAR1 Deficiency. So the IFNAR1 is the interferon alpha receptor 1. It's a receptor for interferon on the surface of cells. Interferon alpha 1 receptor deficiency in otherwise healthy patients with adverse reaction to measles and yellow fever live vaccines. And
I can do authors and affiliations if you
go
want.
ahead. They're
I'll be
huge,
a little while.
Huge.
So we have 5 co-first authors and 8 co-senior authors, and the co-level authors are getting a little out of control, folks. But anyway, here we go. The co-first authors are Nicolas Hernandez, Georgia Bucciol, Lynn Moens, Jeremy Le Pen, and Mohamed Charoui. The co-senior authors are Reynaldo de Menezes Martins, Isabel Mates, Qian Zhang, Margaret MacDonald, Charles Rice, Jean-Laurent Casanova, Emmanuel Zhongguai, and Xavier Bossoit. And they are from... Can I just say they're from everywhere? No, I'll do the affiliations. They are from everywhere, though. Rockefeller University in New York, KU Leuven in Belgium, Sina Medical Complex in Avaz, Iran, Federal University of Rio de Janeiro, University of Medical Science School of Medicine in Bushehr, Iran, Institute for Research in Fundamental Sciences in Tehran, Zanjan University of Medical Sciences in Zanjan, Iran, Necmettin Erbakan University, Mera Medical Faculty in Konya, Turkey, Paris 13 University in Bondy, France, Necker Hospital for Sick Children in Paris, Paris Descartes University, Imagine Institute in Paris, Institut National de la Santé et de la Recherche Médicale in Paris, Ministry of Health in Rio de Janeiro, New York Medical College in Valhalla, New York, Hudson Institute of Medical Research in Clayton, Victoria, Australia, Sidra Medicine, Doha, Qatar, Hamad bin Khalifa University in Doha, University Hospitals Leuven, the Howard Hughes Medical Institute in New York. And I think that's all
Wow.
of them.
Good job.
It's impressive.
Yes,
That
this
is.
is a big team, and it'll become clear why we have some of these countries in here, and then it's just a big collaboration.
So here's a factoid that I didn't know. There are 11 attenuated, this is of 2019, attenuated viral vaccines in use worldwide. So these are the infectious ones. So let's see if between the three of us we can get them. I know OPV, poliovirus, MMR, yellow fever, because they're in this paper.
Flu mist.
Flu mist, that's 4. I guess there's a rotavirus vaccine
Yeah.
in here.
MMR, we said varicella. Did
Yeah.
you guys say chickenpox?
Oh, wait.
Yeah, the original varicella vaccine,
yeah.
varicella, right?
Well, the oral, you said oral polio, right?
I said oral polio. So up to 6 now.
Dengue. Isn't there a dengue vaccine?
Yes, it's based
Yes.
on yellow fever backbone, right? So that's
okay.
7.
Smallpox vaccine.
The smallpox vaccine, 8.
First.
3
Isn't
more.
Isn't there a...
Japanese
it's also
encephalitis.
yeah,
virus.
Japanese encephalitis. Exactly.
9.
Did you guys say Shingrix also?
No.
Like-
So that's
Isn't
just a
Shingrix?
subunit, though. You said
Is
we
it?
said the
But
varicella.
there was an older...
Isn't
Oh,
there
you
a
said
zoster?
varicella. Yeah.
Yeah,
The Zoster
But
vax.
aren't they 2 vaccines?
Yes, the older one, the Zostavax was a live vaccine.
right.
And
So
you
we're
said
at the
that.
9. We're
You said
9.
varicella.
We have 2 more that we're missing here.
What about? Well, it doesn't really exist anymore. But the smallpox vaccine,
we
listed that.
We got that.
we did. Okay.
I have to cheat by looking at.
Wait,
Yeah.
is BCG considered?
And it's not viral, right?
It's not viral though. It's true. It's
No, they
bacterial,
said
but
they limited it to
Protected
They
viral.
limited it to
Okay.
viral.
okay. Then I think that's everything now.
Well, we
I
got
don't
9,
know.
and they said they're 11.
They
so
say
let
they're
me
11.
let me go to my vaccine lecture here, where I have a list of currently licensed.
We said MMR, right?
Yep.
Hopefully,
We said
we got
that's
that.
number 1.
Said MMR. So let's see here. I have a nice table in the lecture.
Yeah, we managed to get MMR and YFV, which is relevant
Yes.
to this paper.
That's right.
So
You said flu mist.
vaccines, that flu mist. Yeah, here we go.
Mm.
So, adenovirus for military recruits, right?
Oh, I didn't even know that one.
Yeah, that's a little
That's
bit
10.
obscure because only military recruits get
Yeah.
it. Nobody else gets it. Let's see. We did flu mist. Japanese encephalitis is inactivated virus.
Oh, really?
So we're back down to 9. I got one wrong.
Well, maybe the 3 components of MMR count
Maybe
separately.
they count separately.
Oh, then, yeah. Then we're
Yeah.
there. 10, 11. 2
Yeah.
more.
So that's my list doesn't have anything else, and there were no
That's probably it. MMR is 3.
attenuated.
Should I ask
COVID.
ChatGPT?
You could. What are the 11? Blah blah blah. Yeah, you could do
Yeah.
that.
So all of these live attenuated vaccines, there are the ones that are in widespread use are excellent vaccines, but all of them have at least been suspected to have some kind of rare, serious complications. They can have OPVs, obviously can cause polio.
Yeah.
Some of the others can cause other systemic disease when the virus gets out of control. So that's what they're looking at in this paper.
And so this has been studied before. So we know, for example, that MMR, the measles part of MMR, can cause disseminated infections in people who have problems with IFNAR2. So inborn errors mean you're born with a polymorphism and that causes loss of function of these components. And they say vaccine strain mumps infections are very rare, with only one life-threatening case in a patient with RAG1 deficiency. So the RAG determines the production of T and B cells. So people who have deficiencies in a gene called IRF7, which is like a master regulator of interferons. It controls amplification of type 1 and type 3 interferons. They suffer from ill-defined adverse reactions to MMR. And most cases of severe MMR aren't explained. And that's one of the things they want to do. And also, none of the life-threatening adverse reactions to live vaccines for rubella, yellow fever, influenza, varicella, vaccinia, or Hep A. Oh, there's Hep A. That's one we
Hep
forgot.
A.
Have been explained. None of these have been explained genetically. So that's what they did in this paper, is to look at some patients with severe disease caused by MMR and yellow fever vaccines.
And I think it's, especially with vaccine hesitancy, I mean, this paper was 6 years ago, and I feel like it's only grown since then. It's important to highlight genetic predispositions. Like, people can have genetic deficiencies, X, Y, Z, that we're going to talk about, that are the cause of that adverse reaction, not necessarily that the vaccine is inherently dangerous. So the more papers like this, the better, I think, especially for vaccine hesitants, just to explain some of those adverse reactions. It's not that, the vaccine itself is inherently evil or going to
So
kill
that's
you.
actually why I picked this, is because, RFK Jr. says measles vaccine kills people all the time. And it doesn't. This one is a rare event. I don't know what the frequency is. They don't actually give numbers. But this is actually the first death due to a measles vaccine that I've ever heard about. And it occurs in people with immunodeficiency. So we'll talk later about what to do. So patient 1.
The patients in this study actually don't die.
One of them.
One of the siblings.
One of the siblings died.
Right. There's a sibling who we think probably had the same.
Yeah.
Yeah.
All right. Patient 1 was born in Iran to co-sanguineous parents. Not a good idea, folks. Okay?
Don't marry your cousins.
Don't marry your cousin. We know from many, many years of experience it's not a good idea. So at 1 year of age, had disseminated vaccine-strain measles. Must have been vaccinated. 10 days after inoculation with MMR, had generalized exanthem, rash, fever, neurological symptoms, which look like encephalitis and confirmed by CT. PCR for measles positive in the blood and CSF. No other medical history before that. Of course, 1 year old. Shouldn't usually. Now this patient 1 is 9. and is fine and has had no other problems. You're going to see this patient has an interesting defect, and I don't know why it doesn't have
It doesn't.
any.
It didn't
It's
affect
surprising.
anything else, apparently.
No. Patient
Somehow.
1 had a younger sibling who died 4 weeks after a routine MMR at age 1 year, but they didn't have any material, so they couldn't do any genetic analysis. Patient 2, born to smart parents, this is a Brazilian girl, 14-year-old, to non-consanguineous parents.
Okay, smart parents. There are cultural issues here that may have led
Obviously,
to the
yes.
particular outcome. And I will say that patient 1's mother terminated a third pregnancy after they found they genetically identified a variant that we're going to talk about in this paper.
Yeah. Which,
so
okay. But she probably didn't have to, because the other girl is okay, and so you just don't give them MMR vaccine. I don't know what you're going to do otherwise. But so at age 12, she was fine up to age 12. Then she had fever, hypotension, vomiting, and lethargy. A week after yellow fever vaccination.
Right, we're on the Brazilian patient now.
Yeah. So yellow fever is endemic down there. So
I guess it's a
she got
routine.
very, very sick. Renal and hepatic dysfunction, need to be intubated, mechanical ventilation. Yellow fever RNA in the blood and by PCR and sequencing showed it was the vaccine strain.
Mm-hmm.
She recovered. She's now healthy, and she had received 2 MMRs at 12 and 16 months of age, much younger, and other vaccines,
with them.
And poliovirus vaccine and BCG without any problems. Okay,
Which is crazy considering what we know now.
Yeah.
Like after I guess we can discuss at the end. Like, how did they tolerate these other vaccines,
how
lab
is this
attenuated vaccines?
So specific, yeah.
Crazy.
So they they made DNA from these patient cells, and they do whole exome sequencing. Okay, to get this genome sequence and find out what could be wrong here. And interestingly, so they look at the percent homozygosity of different genes, 2.9% for patient 1, which is high, and 0.25%, which is normal because it's cosanguineous and non-cosanguineous, right? They then look through, there are lots of changes, as you might imagine, and these have to be filtered out. They end up in patient 1, a homozygous mutation in IFNAR1. Okay, IFNAR1, the interferon alpha receptor 1. In patient 2, 11 variants in 6 genes initially, including 2 variants in IFNAR1 and some other genes. And these apparently are novel at the time. They didn't find them in any databases. So a little bit more on IFNAR1. So the IFNAR1 gene and the IFNAR2 gene make 2 proteins that serve as a receptor for type 1 interferon. So it's a heterodimer, right? And these mutations in the gene for IFNAR1 are bad. They're predicted to be bad, and we're going to go through some studies to actually confirm that. They disrupt an essential splice site or create a premature stop codon. So this is all found in the whole exome sequencing of patient DNA. Then they go back to the patients and their relatives and sequence the gene, and they confirm that the mutations are present. And in particular, patient 2, it's heterozygous because non-cosanguineous parents. No DNA from that child who died, unfortunately. So
and
basically,
the IFNAR1 receptor is basically on all cells, right? I think it's ubiquitous on almost every cell type.
Yes.
And but these cells can make interferon, type 1 interferons. It's just the receptor that is deficient in this case. Yeah, just to
you need
clarify.
IFNAR1 and 2. So the patient's made IFNAR2, but it's not enough on its own. So basically, these are rare mutations, right? They're rare polymorphisms. And so it looks like these 2 patients had inborn errors of immunity, autosomal recessive IFNAR1 deficiency, so AR IFNAR1 they call it. So then they go ahead and look at these mutations and see what they do. So in one of them, there are mutations that are predicted to lead to aberrantly spliced mRNAs. And so they amplify. So they have, they take some cells from the patients and they immortalize them so they can grow them forever in the lab and study them, right? And so they have immortalized cells and they can amplify the IFNAR1 mRNA as DNA from these cells, and they can sequence it, and they find messed up splicing, which basically causes a frame shift and a premature stop codon. And so the protein is deleted, and they say, not likely going to work, but we'll check it. And they do this for all the mutant alleles and show that they're not likely to be functional. So then they look at patient cells and they do PCR for IFNAR1, and there's not a lot of mRNA. Remember, this mRNA has got a premature stop codon, and in cells, there's a mechanism to recognize mRNAs that are messed up. So premature stop codons or other things. It's called nonsense mediated decay. That's what we're undergoing in the U.S. right now. Nonsense mediated decay.
That should be the title of our episode.
Because it's a defective mRNA, and so there's less of it in the cell, right? So then they say, is there any IFNAR1 protein? So they do extract protein from the cells, and they do a method called Western blotting, where you can use antibodies to look for the protein. 0 IFNAR1. So they don't detect any IFNAR1 in cells from the patients. And IFNAR2 is normal. It's on the surface, in fact, of cells. But
it's just waiting there for the IFNAR1 so they can get to work.
Say, where are you? Come on, we have a reservation here. Need to get going. So that was normal. So then what about, so when interferon binds the interferon receptor, it triggers a signaling cascade that results in the production of interferon stimulated genes in the nucleus. And so they take these cells in culture and they add interferon to them. And one of the intermediates in the cascade, there are two called STAT1 and STAT2. They're supposed to be phosphorylated as part of the signaling, and they're not phosphorylated at all. And there's no ISG expression. They look at a couple of ISGs, but when you add the interferon alpha or beta, and they share the receptor, alpha and beta interferon share the type 1 receptor, no induction. But if you add interferon gamma, which uses a different receptor, everything is okay. They make ISGs.
So the type 1 interferon response in these cells is broken.
It's broken. Major defects in signaling and induction. And other people have described deficiencies in STAT1, STAT2, IRF9, another regulator, IFNAR2. So this is IFNAR1 deficiency.
Yeah, they show in the data all of these other patients that if you look at the figures, you can see them like IFNAR2 deficient people, STAT1, STAT2. So I'm guessing this is part of this huge collaboration. It's probably just because they needed all of these very rare samples of people that are like IFNAR or IRF7 deficient, which is not that common, I'm sure.
Yeah, all of these are very rare, but so it looks imposing when you see all of them on a figure and you go, oh my gosh, but extremely rare. I'd like to know the frequency, but I just don't know. It may be hard to know because unless you get sick, we don't do any screening, right?
It's true.
I don't think 23andMe even looks at this stuff.
No, I don't think so. Unless it's a severe immunodeficiency, you would never really know.
Yeah.
Is it 23andMe that's going under?
Yeah, they were just bought by Regeneron.
Oh, okay.
So Regeneron wants to continue the service, but also mine data.
Mine all the data. Yeah.
So,
of course.
At one point, 23andMe was valued at $6 billion. Regeneron bought it for $225 million. What a deal.
That's
quite a come down.
Yeah. Okay, so finally, can these cells control virus infections? Right? That's a nice phenotypic
Yeah.
assay there. So they have these immortalized cells, they're fibroblasts, and they infect them with, they treat the cells with interferon alpha and then infect with VSV, which is what you often use to titrate interferon. Now, healthy control cells, when you pretreat with interferon alpha, controls VSV infection, but not the patient cells. The virus infects and goes nuts because the interferon is not doing anything.
Hmm.
And they say this is also seen in IFNAR2, STAT1, STAT2, and IRF9 deficient cells. And then they infect these cells with the vaccine strain for yellow fever virus, right? And same results. So the interferon deficient cells can't control infection with the vaccine virus strain. But, and this is how complicated it is, IRF7 deficient cells have no problem controlling yellow fever vaccine virus reproduction. So when, adding exogenous interferon. So there's some nuances going on here that we don't fully understand. So then they want to confirm that the defective IFNAR1 is doing this, so they put the gene back in. So they have a vector and they put IFNAR1 or IFNAR2 in, or empty vector, and only IFNAR1 is rescuing the defect in IFNAR expression, responses to interferon, and responses to virus infection. So if you put IFNAR1 back in, now they can control virus infection in the presence of interferon.
Mm.
And that goes for VSV, measles virus, and yellow fever virus. Then they infect these patient cells with influenza virus and herpes simplex virus. And they say these patients had been exposed to these viruses but didn't develop severe disease. The cells support higher levels of replication than healthy controls for both viruses. But I don't, that's all they say, because these kids never got sick with herpes or influenza virus. They also looked at another yellow fever vaccine strain. Well, actually, it's a wild type strain, yellow fever, a CB, and Zika viruses. And both patients were unable to control viral replication of those viruses. I guess that includes herpes and influenza virus when adding exogenous interferon beta. So IFNAR deficiency gives you a broad defect in innate immunity, fibroblast intrinsic immunity, they call it, or innate immunity. All right, so IFNAR is defective. What about, is there any problem with interferon production? So they look, they quantify the production of interferon beta, which will bind this receptor. And they have actually higher amounts of beta in IFNAR deficient cells after virus infection. So the defect is signaling, not the production of interferon.
Yeah.
Right. So it's cell intrinsic, which I would call innate immunity to type 1 interferon immunity, VSV, yellow fever vaccine strain, influenza A virus, herpes simplex virus, and measles vaccine strain is abolished in patient cells. And that suggests why these kids had problems after vaccination. And
it doesn't explain why they didn't have problems after other vaccines.
It doesn't. No,
or illnesses or all the rest of everything.
Just
because
living
the kids
in
are
life.
fine other than these specific episodes.
So this is interesting. They go through other findings and mutations in genes encoding like STAT1 and STAT2, and they say that in humans, the data differ from work done in mice, where STAT1 is dispensable for defense against both wild type and attenuated measles virus. So they say this suggests a set of genes jointly regulated by IFNAR1, 2, STAT1, and 2, and maybe IRF7 and 9 are critical for control, but we don't really know, right? So then there is a discussion here. Okay, this is what we want to do. So these two patients they're now 9 and 14 years old. They have not had clinically significant viral infections other than the ones reported here. And they said there's another patient with combined partial IFNAR1 and complete interferon gamma receptor 2 deficiencies, a different interferon. Got CMV infection in addition to BCG disease. So it's pretty complicated. And they say, okay, 3 hypotheses could explain the antiviral defense in 2 patients, even though they're lacking type 1 interferon receptor. First is that the splice allele is leaky. Maybe it's not defective in all cell types. Or maybe IFNAR is redundant. And maybe in some cell types, there's not a deficiency. And the third is that IFNAR and all type 1 interferons are redundant for immunity against most viruses, at least in certain conditions of infection.
And the first 2 stem from the fact that they only looked at these immortalized cells that they could get from the patients.
Yeah.
And you can't really take all types of cell samples from a patient and get a real view of what's going on in other types that might be relevant. The third one is just kind of, well, okay, maybe type 1 interferon's just not that important.
Yeah. Well, I mean, they say...
which is crazy to say, but...
They say it's probably relevant that patients with IFNAR2, STAT2, or IRF9 deficiency, which impair responses to type 1 interferons and type 3, that's for STAT2 and IRF9, are apparently also normally resistant to a broad range of viruses, including CMV, and have survived until 5 to 11 years of age. Whereas all patients with complete STAT1 deficiency, which impairs type 1, 2, and 3 interferon responses, they're dead by 2 years of various viral infections. So if you get rid of all 3 interferon types, you're really in big trouble. But one, you can deal with it. The system is redundant to a certain extent.
We have IFNAR knockout mice, and they live in an SPF room, which still has viruses. There's just like a few things that we test for, and they survive. I mean, I'm not using them. Someone else in my lab does, but they seem to be doing okay. They don't just like randomly die. So, and they're obviously in a dirty.
That brings up an interesting historical tidbit. So we made these polio receptor transgenic mice years ago in the lab, right? Put the human polio receptor gene into mice, you can infect them and they get paralyzed. Mm-hmm. The virus only seems to reproduce in CNS, right? If you then cross them to IFNAR lacking mice, which you're just talking about, you infect them with polio, it infects everywhere. They don't die, but the virus is reproducing everywhere. The implication is that the interferon is protecting most organs from infection except the CNS for whatever reason. So what do we do about this? They say here we should develop safer vaccines for people with these inborn errors.
Well, how are you supposed to know that they have these inborn errors?
Well, I know that's a problem, unless we screened everyone, right?
Yeah, so I mean, the thing is,
sequencing.
Developing a whole new vaccine to replace something like MMR, which is a critical vaccine that's given globally, rolling that out to account for some very, very rare cases where it can cause these problems may not be the best use of resources.
No, I don't think so.
But as we get, cheaper and cheaper genetic testing capabilities, I could certainly see something where, you already test a newborn's blood for a bunch of different things. Maybe you do a quick screen for a panel of rare but potentially serious mutations, and you say, oh, by the way, make sure your kid doesn't get the MMR vaccine, and this is a legitimate medical exemption because they have this mutation.
Well, I mean, especially in areas that have high consanguinity, areas that because not only just like this mutation in itself, but lots of heritable immunodeficiencies are increased in people that are from consanguineous, in a consanguineous parents. So even just in that case, like here in Quebec, actually, Quebec has a very high rate of consanguinity and heritable immunodeficiencies, primary immunodeficiencies, actually. So in that case, in areas where the researchers, like the universities in that region that know that they have a lot of immunodeficiencies, maybe it's more prevalent and maybe more interesting to do it there specifically, like screen for that.
Sure.
So,
There are also populations that it's not what we would normally think of as consanguinity, but people who marry within a particular group.
yeah,
You know,
it's like a
you're
small
not cousins,
town that
but if you
everyone
go
gets
back
married.
far enough, they're pretty
Yeah.
closely related, and then you get a population that can have similar sorts of problems.
Mm-hmm.
Yeah, I mean, if we had inactivated vaccines, it would get rid of this problem, right?
Sure, inactivated vaccines or subunit mRNA vaccines or other technologies like that where you don't have replicating virus.
Well, that's a trend for polio. We should be switching to a
Yes.
non-infectious vaccine at some point in the future. And so that would eliminate that. But there are many others, right? Yellow fever vaccine is not going to be replaced. But I saw a paper this week on an antiviral for yellow fever. So that could be useful if a kid gets disseminated yellow fever after vaccination, you could treat with an antiviral.
Yeah.
And save their life, right?
Hmm.
So that may be another approach, because developing vaccines is probably not going to happen if it's so rare, right?
Yeah.
Anyway, so that's measles vaccine does not kill a lot of people, as RFK Jr. claims.
No.
This is one case that I know of. This is the only case that I know of, right? Because the guy who was on TWiV and talked about measles. Oh, here he is, Adam Ratner. I sent him this paper because he says there have been no deaths from measles vaccine. So I sent, you probably should just say there has only been one from now on. And the anti-vaxxers have not found this because they don't read.
Right. No, they
Of course
selectively mine the literature for any kind of, little thing that can.
Do you even think they mine the literature? I think that's also very generous.
I agree.
That's
Angela. I was
very
just going to say the
generous.
same thing. That's giving them a lot of
They
credit.
go fishing in the literature.
They go
give
on
a lot of-
Facebook and they
Yes.
receive what the algorithm pushes them. I know
Yeah.
now there's no news on Facebook. Okay,
I
other
agree.
social media,
I
Twitter,
agree.
or sorry, X.
Zitter. Yeah.
Exactly.
Okay, let's do some email. We have some time. Angela, can you take the first one?
Yes, let me open that up. Ben. Okay, Ben writes, Dear Twixters, you don't have to read this on the show if it doesn't suit. Well, here we are. Writing to you from Perth, Australia, where the rains finally started kicking in, way too late for the season. It's going to be cold. It's going to be a cold night for Perth with 6 degrees Celsius. A few days ago, I was incredibly surprised to find a package with a TWiV shirt. Thank you so much. I guess that's for my Patreon contributions, which I started years ago, so I totally didn't expect this to show up now. Coincidentally, I had to pause the donations because of a change in financial circumstances. We'll start again when we're back on track. I am flabbergasted how quickly things have turned sour in the US for science, but also in general. Your stories are jaw-dropping at times. The impact is also felt down under, where it is argued that the anti-Trump effect has led to an unexpected swing towards less conservative government at the recent federal elections. We were on the verge of getting back to the science-denying circus of malignantly incompetent clowns that you are suffering from. So thanks in a way for the bad example. Thanks for all you do and hang in there. Keep up the fight, Ben.
So
people email me too, even from Spain where I used to live, and they're like,
Yeah.
what's going on in North America? And I'm like, I don't know, I'm in Canada.
So when we started Patreon many years ago, I had put in, if you donate this much a month, you get a mug or a T-shirt or something else. And I never had time to send any of this out. But now we have Kim, our social media person. She's taken on the task of doing this and shipping out many, many dozens of things. So I'm glad
She's great.
you got
Excellent.
it. And now this is good because people use it and then other people will see it and ask what it is and maybe get new subscribers, right?
Yeah.
Alan?
Okay, Bree writes, Hi again. New email with new job, but same bumbling biochemist here. Following up on Rich's suggestion of Wikipedia, I'd like to recommend listeners get actively involved with Wikipedia through Wikipedia editing. I started editing in grad school, inspired by Jess Wade, who wrote tons of articles on female scientists. I followed suit and have since expanded to edit whatever I come across that needs help. The latest, I'm editing your email as we read it. The latest was a few days ago when I was helping one of my students look up something about cortisol on Google, and Google's AI cited Wikipedia, which was inaccurate. So I edited the Wikipedia article to fix it and got to teach my students about using caution and employing critical thinking while using AI, as well as introducing them to Wikipedia editing. I have a blog article with resources and links, etc., gives a link, and Wikipedia has training as well, gives another link. Whether you're wanting to create new articles, build up existing ones, or just fix typos, there's plenty of work to do, so I encourage listeners to find what suits them best. The power of Wikipedia comes from all of us. Thanks again for all you do. The bumbling biochemist, Brianna, who is an incoming professor of biochemistry and chemistry at Loyola Marymount. Congratulations on the new job, Bri.
Congrats.
Cool.
Yeah, it's good to fix Wikipedia.
Yeah.
Mm-hmm. I think it's gotten better over the last 2-3 years. I remember 10 years ago, Wikipedia was crazy. You couldn't believe half the stuff on there,
Yeah.
but people fact check it quite a lot now. So I think it's come along.
And I think they've improved their procedures. They've got a continuously evolving process.
I'm a contributor. I think it's important to support them.
To Wikipedia, definitely.
Yeah.
All right, we have two from the same Chris. His second one is short, so I'll take both. Chris writes, immediately when I saw Dr. Griffin from the lower camera position, I wanted to tell you how great the shot was. Dr. Griffin looked so handsome with the dark background from that position. Striking. So nicely done, sir. So Daniel was in the studio last week for clinical update, and I set up the shot. And I'm not very good at setting up shots, right? I just go with what I feel. And I had a camera on Daniel and then another camera on both of us. I figured that would be good for the editor. So I'm glad you liked it.
Yeah.
I tried to, so not too much space above the head.
Yep.
And not a lot. I don't want to see legs really and feet. Certainly not feet. You don't need to. So
It's
waist
true.
up. And these lenses have a zoom, so that's good. Two other observations, if I may. First, a coffee table in front of your two red chairs might be more comfortable for you. It would also soften the scene somewhat, facilitating your brilliant conversational style. That's why I wanted to read this letter. Right? Coffee table. It's no room, really. So we have this glass table in between us. I think that's fine. And finally, I'm wondering how I could make hexagon signs with the logos of all your programs, one for each panel. So behind us, we have these hexagon panels. They're actually sound deadening panels in different colors. And they kind of look like viruses, right? It would advertise your other podcasts while adding a bit of complexity to the eye, stimulating learning and memory. I think a Velcro fastener would be simplest. Let me think more about it. That's it. That's an interesting idea because often you have logos in the background of people who are being interviewed, right? This is Chris in Toronto. I'm 58F. I'm old enough not to be comfortable with metric. Well, I bet you're not older than me. And I love metric because I'm a scientist and scientists use metric, right?
So much better.
Yeah, but it's confusing because, everything around here in the U.S. is all in English measure. And I sometimes think in nautical miles too. And it's just,
So, people ask me what the temperature is. I give them Celsius and they, well, what is that? I said, well, figure it out. It's not that hard.
Or also just use your phone in 2 seconds now. You can know.
Yes, of course. My phone, of course, is in Celsius. My car is in Celsius. The only thing I don't have is the heating and cooling in my home. There's no way to change the thermostat, or else I'd have that in Celsius too. Just put it at 20 or 22 and you're all set. Probably 22. 20 is too cool.
20 is a little chilly.
20 is good for warmth, though, right? That's enough. 68 is enough.
Yeah.
All right. And the second from Chris. Robert Sapolsky, in minute 26 of the podcast, fully explains The Proven Science, Debunking Vaccines as a Cause of Autism versus RFK. So well said. So he provides a link to a YouTube video. So check that out.
Did he send those the same day, or was there like quite a big, do you know? Is this like
Not
second
a big one.
thought?
It's just a few days.
Oh, okay.
Maybe a day. I don't know.
Okay.
Okay, Angela.
John writes, colleagues, in your discussion of the replicating mRNA vaccine, you talked about vehicles used to deliver the mRNA. During this discussion, mention was made that Moderna's SARS-CoV-2 vaccine was more reactogenic. While this is true, I believe it is more reactogenic because of the dose at 100 micrograms for Moderna's vaccine versus Pfizer's 30 micrograms. Love your TWiV episodes. Thanks so much, John.
You can take the next one, too.
Alex writes, Dear Professor Racaniello, I recently came across a perceptive comment by John Dolan on his Radio War Nerd podcast on the psychology of social phenomena like the lab leak theory. The episode is on an indigenous Amazonian tribes encounter with Westerners in the 1960s. The context of Dolan's comment is that this tribe suffered a severe flu outbreak on first contact because they had no natural immunity. To this day, the folklore in this tribe is that Westerners poisoned them or cast some kind of spell. Dolan remarks, I've read this interesting generalization that in many groups that have not evolved for themselves the idea of germ theory, there isn't a very strong idea of natural death. Every death has to be attributed to someone's malice. And I think we saw it again when COVID happened. It can't be just one of those bad luck plagues. Who do we blame? I mean, is it Chinese labs or the Democrats or what? See, and then he has a link to podbean.com for an hour and 15 minutes into the episode. Around an hour and 15 minutes into the episode, I guess, is this little excerpt. Kind regards, Alex.
Yeah, I think this is, so it originated as a political issue, but then people probably have this sense that it's, I think that makes sense to me.
And here in the U.S., we have a long-standing national tradition of finding somebody to blame for everything.
That's right.
Yeah.
Speaking to you from Massachusetts, where we had witch trials.
I had this long email today to TWiV, which I'm not reading, because it's someone who disagrees with everything we've talked about the nature origin of SARS-CoV-2. And right at the onset, this person says, I just think it's more likely it came from a lab. What does that have to do with anything?
Matter? Yeah.
Who cares?
I have a feeling.
Yeah, that's exactly how the reasoning of that works. Is you start with the conclusion and then you seek out evidence for that conclusion, which of course is exactly the reverse of how science works.
It's too complicated the way you're proposing. Well, SARS-1 came up in that complicated way. It does happen. Give me a break.
And it's not that complicated.
It's not that complicated.
Viruses in wildlife hanging out in animals. Potential intermediate host spilling into a human that's around them. That's it. It's not that crazy.
Alan, can you take the next two?
Sure. Anthony writes, another show with your vet friend from the Bronx Zoo. Diagnosis of tuberculosis in 3 zoo elephants and a human contact. Oregon 2013. This is MMWR. Oh, that could be cool for TWiM, right?
Mm-hmm.
Yeah, so Paul Cowley was the director of the New York Wildlife Society, which manages all the zoos in the New York area, including the aquarium and the Bronx Zoo. So we did a TWiP there, and this is who he's talking about.
Cool. Interesting.
Angela, did you know TB infects elephants?
I did. TB infects, there's lots of different mycobacteria that can affect a lot of species, actually lots of, as we know.
Wow.
But...
Yeah. Okay. Abigail writes, hello, TWiV team. There was an excellent letter sent in on TWiV 1213, encouraging a microbe TV attempt to extend listening and viewing audiences to the lay population. Instead of creating a new resource from the ground up, though, why not utilize pre-existing resources? My pick of the week is the podcast Ologies by Ali Ward, which is available on all streaming platforms, but I prefer to use Spotify. Ali is a science communicator who interviews ologists, or experts in their fields. She also incorporates listener questions, allowing for an element of interaction. Ologies spans a broad array of subjects, but I've selected 3 episodes below that TWiV listeners might be interested in right off the bat. The most recent episode on long COVID was released in March 2025. The 2 older episodes were released during the heart of lockdown. Not only are these episodes and this podcast fun listens, but they are an easy way to provide information to the general public. Allie asks very pointed questions and adds in plenty of asides to elucidate more complex topics. I like to keep certain episodes in my back pocket so that I can forward them to anyone I think might benefit. There is also a spin-off podcast that is classroom-friendly for a younger population called Smologies. Nice. And links to episodes on long COVID, what to do during a pandemic, and vaccine infodemiology.
I love ologies so much. I have to say, Allie Ward, love her. I listen to it all the time because she has like random ologists on about like volcanoes, dolphins, like literally experts on anything. And the Smologies is actually, it's only because she swears a lot. So Smologies is important because there's no swearing. So she's very like, candid, and there's a lot of swearing from her and her guest. So Smologies, if you have like children in the car, if you're listening, is definitely more friendly if you're sensitive to swearing.
when I have a child in the car, they'd better not be sensitive to swearing just because of, Well, anyway. And there's a PS. I've used plenty of the papers on TWiV for my PhD program's weekly journal club. They've sparked great conversations. Cool. And Abby is a PhD candidate in microbiology and immunology at Dartmouth College.
Okay. It's my turn, right? Yeah. Anonymous writes, the news below has not really gotten out there, but lost in the plethora of bad news on the research health front, so I wanted to pass it on. So this is the Walter Reed Biosystematics Unit update. The Defense Health Agency has announced the formal dissolution of the Walter Reed Biosystematics Unit. WRBU had been a partnership between the Department of Defense and the Entomology Department of the Smithsonian for more than 60 years, undertaking cutting-edge research to provide actionable entomological intelligence tools and products that best assess global vector-borne disease risk. Their work helped ensure the health and safety of our troops and civilians stationed in locations around the world. The program's manager, Dr. Yvonne Marie Linton, will remain a member of the National Museum of Natural History entomology staff, where she's committed to continuing this impactful research, combining cutting-edge genomics and collections-based biodiversity discovery. NMNH will continue to steward the national collection of biting and blood-sucking flies. Did you know we had a national collection?
Did not.
Including its extensive collection of mosquitoes to ensure it is of maximum benefit to the American public and the global research community.
I'm sure Dixon would know. He would have known for sure.
Yeah. This is just another example of people cutting things they don't know anything about.
Yeah.
Mm-hmm.
They just want to save money so they can give it to wealthy people.
Yeah.
Unbelievable. Let's do one more round. Angela.
Marie writes, I've been listening to your podcast since 2020. I recently discovered with self-reflection that I am comforted when I hear your brief theme music. It's probably a response similar to Pavlov's dog. The music causes a bodily response of, these scientists have my back. Even with the government being crazy and dumb, these intelligent individuals care about me and my family. In this horrendous time, I am grateful for all your continued work producing the podcast episodes. Thank you tremendously, Marie.
Oh, that's very nice. Thank you. We do have your back, right?
We do.
Yeah.
Alan.
Mike writes, in case you've not seen it, U.S. attorneys are sending out, quote, vaguely threatening letters to scientific journals about what they publish. And provides a link to an Instagram post from Peter Mazzone, editor-in-chief of Chest Journal. This is a major medical journal. Dear Dr. Mazzone, United States Attorney for the District of Columbia, I receive frequent requests for information and clarification. I take these requests seriously and act on them with letters like this one you are receiving. It has been brought to my attention that more and more journals and publications like Chest Journal are conceding that they are partisans in various scientific debates. That is, that they have a position for which they are advocating either due to advertisement under postal code or sponsorship under relevant fraud regulations. The public has certain expectations and you have certain responsibilities. Would you please answer these questions? And then gives a list of leading questions that are somewhat accusatory. How do you assess the role played by government officials and funding organizations like the National Institutes of Health in the development of submitted articles, as an example? And then goes on to say, are there new norms being developed and offered? I look forward to and appreciate your cooperation with my letter of inquiry after request. Please respond by May 2nd, 2025, etc. And that's from
I should have
Edward
edited
Martin,
this.
U.S.
ChatGPT
Attorney for District of Columbia. Yeah.
You could have had like a little ChatGPT edit before sending that.
Yes. And then
Pretty bad.
So Mike's letter says, Dr. Zachary Rubin is a pediatric allergist, clinical immunologist with an Instagram page dedicated to countering public misconceptions. I noticed him speaking out about the measles outbreak in Texas. He is a bow tie aficionado like Dr. Daniel Griffin. I heartily endorse his channel, another voice of light in these dark times. And that's where this letter was posted. So now threatening journals that publish facts.
Alan, what is this stuff about advocating their partisans in various debates? That's not true, is it?
No. This is about they have criticized misinformation coming out, or they've said things that do not agree with misinformation and disinformation coming out of the White House is basically what's going on here.
I see. Okay, one more here. Richard writes, hello, TWiV gang. First, let me thank you for all the work you've done with SciComm. I've been an avid listener from almost the beginning. I think I started on episode 10 back in my graduate school days in Boston. There's a chance Brianne may have been a classmate of mine. My PhD is in virology, examining how amino acid substitution in HIV integrase affect viral replication, ranging from enzymatic activity to integrase to effects on genome targeting. As for why I am writing this, in reference to the recent discussion of novel bat coronavirus research in Wuhan and which biosecurity level it should have been performed. Although the work was performed at BSL-2+, the discussion focused mainly on whether it should be BSL-3 as opposed to 2. One has to note there's a distinct difference in BSL-2 and 2+. Plus designation, as I've come to understand, was created for HIV-1 research. This was to allow for a heightened level of security for an agent that is likely BSL-2. This means that researchers would use BSL-3 standard operating protocols, but adapt them for the agent. Operationally, this meant PPE would be Tyvek suit, Tyvek boots, face shield, double gloves, but shower in and out was not required. Experiments have to be performed in a negative pressurized environment with minimal sharps. Contents coming out of a biosafety cabinet have to be sterilized before it can leave, and all waste has to be autoclaved. Access to these environments are restricted to trained personnel. So operationally, for all intents and purposes, the work is done at BSL-3. Obviously, we can debate whether the novel bat agents should be given to BSL-2 or BSL-3. Risk assessment is highly difficult, can go either way. There are those who consider any virus research to be highly dangerous and should not be performed in any way or form. Gradations of this sentiment extend to opponents of wildlife surveillance, gain of function, and dual-use research. The other end, scientists doing whatever they want, though, led to many unfortunate incidents and fantastical stories, such as Jurassic Park 28 days later and The Walking Dead. So although imperfect, the Wuhan Institute decided that the novel coronaviruses were likely BSL-2, but took the precaution to work at BSL-2+. Perhaps they should have worked at 4, or even a dedicated BSL-3, although they did work at its operational equivalent, if for no other reason than the optics of the solution, though from the Chinese perspective, it's the U.S. that should be conducting research more carefully. Anyway, just wanted to clarify the difference between 2 and 2+, as I think that got a little lost in the discussion, and frankly, Richard, I didn't even know there was a BSL-2+.
My understanding is that the BSL-2+ standard is not rigidly defined in the same way as BSL-1, 2, and 3.
Yeah.
Isn't BSL-2 plus also, I guess it's different from BSL-2 plus is like when you have live animals. Like when I infect mice with viruses, it's technically they call it BSL-2 plus, where the mice is a live organism that's replicating a virus is BSL-2 plus. So we have more PPE than if I'm just harvesting their tissues, taking them to the lab and processing them. That's regular BSL-2.
Okay.
Right.
I think at least that's how we define it. There's probably other definitions, but
yeah, that's the issue. And BSL-2 plus, if it is operationally equivalent to BSL-3, then you probably ought to be doing the work at BSL-3, especially if your institute has been in the spotlight the way WIV has been, which was the point I raised. Just, for the appearances.
For the optics, right?
Yes.
As he says.
It's not BSL-3 though. BSL-2 plus and BSL-3 is quite different. We also work in the BSL-3, and that's like different PPE, different negative pressure systems, and like even just the sterilize it, like the moments of like Oxivir, you have to use like accelerated hydrogen peroxide for everything. You have to wait 3 minutes after you spray everything. It's so annoying. We don't have to do that in the BSL-2 plus. I mean, it's annoying like necessarily, obviously, but everything takes 8 times longer in a BSL-3. Whenever I see anyone doing work in a CL3, it takes longer than in a CL4, because in a CL4, you're even more protected. You're just wearing a full positive pressure suit. The CL3 is terrible to work in. Let's
do some picks of the week. Angela, what do you have for us?
What did I choose? Okay, so I chose something that was really cool, I thought. So a paper came out in 2019 where in Cell that was called Mammalian Near Infrared Image Vision Through Injectable and Self-Powered Retinal Nanoantennae. So this was by Yu-Kian Ma et al. And basically they injected nanoparticles, so photoreceptor binding nanoparticles into the retina, I believe, of mice. And it allowed mice to develop infrared vision for up to 10 weeks. And this was like six years ago. So that was really cool because normally mice don't see infrared. So for those people out there that don't know, the only animals, at least avian species that I know of that can see infrared are birds of prey. And I love this factoid. Pigeons can also see infrared. I learned that in vet school. It was the most random thing. Evolutionarily, who knows why? Because they're not flying at like 500 meters to attack a mouse or to kill a mouse, but who knows? So then they took this this research, and where's the other article that I have? So I guess I linked the Nature paper, so a Nature article that came out yesterday, showing that now another group has made contact lenses that, like human contact lenses, that use the same technology with these nanoparticles that can actually convert near infrared light to shorter wavelengths, so to make it visible so humans can see. And they estimate that it'll cost about $200 per pair to make. So maybe they'll sell them for like $1,000, who knows? And that it is slightly blurry. They did say that there's some cons still, that it's slightly blurry, and that they don't actually know how useful these are going to be in real life. Because obviously we have like the goggles that people use like with night vision, which is also infrared. But I think it's interesting. It's really cool that we can do that now on a contact lens. Who knows if that's going to be useful or not, but I thought that that was just really cool that they're now creating contact lenses with infrared lights. And
they
were
can
saying
imagine
that
the military being very interested in this.
Exactly. And even doctors, they were saying that for certain surgeries and things like that, if they needed to see like, I forget what the exact technique they were saying that we would use. Yeah, like with excision of different types of tumors and different techniques that they were using that they would need infrared that potentially. But I'm not sure how useful it'll actually be. I just thought it was really cool that it went from like mouse to human in like 5-6 years. It's pretty fast.
It's not bad.
It's cool. So
neat
That's
stuff. Amazing. I hope those nanoparticles in your eye are okay.
I mean, it's on a contact lens.
Yeah.
So yeah, I guess they've done human trials, because if not, then they wouldn't be offering them. But I don't know if I'd want that. But I think it's cool.
I definitely would not want version 1.0 of it.
Definitely not. I want like 11.0.
Wait for some people to try this out first.
I'll wait a decade, maybe.
Okay, Alan, what do you have for us?
I have a pick from a game development blog that I'm subscribed to, because I've picked up a couple of this guy's very interesting games. But why I'm picking this post is it's about politics. It's about the current situation in the U.S. and how a whole lot of people are kind of despairing about it. And he makes the very persuasive argument that despair works in favor of autocrats, because as soon as you say, well, we can't stop this, a majority voted for this, I guess this is what we deserve. First of all, that's wrong. And secondly, that allows this to continue because the president of the United States does not actually have the power to do a lot of these things. He needs all of the assistance from all of the other politicians and also the people in order to pull this off. And what I'd like people to do, if you don't feel like reading the whole article, is just scroll down to the graphic, which is a statistical analysis of U.S. presidential election results for the past 50 years, going back to 1976, and who won what percentage of the vote. It's a very simple concept, but it is very enlightening, and I found it surprising to look at who has won every election except one in the past 50 years, and that is did not vote.
Yep.
So you go back to Reagan, who's, thought of as this unifier who won landslide elections. 48% and 47% of Americans in the two times he was elected didn't vote for him, didn't vote for anybody. And then, 22% voted for his opponents. So you get up to the only election where this hasn't been the case was 2020. Joe Biden won an actual majority for the only time in 50 years.
And barely.
Just barely, yes, but he did. And then 2024, 31.03% voted for Harris, 31.97% voted for Trump, 36.37% didn't vote. So over 2/3 of Americans did not ask for this. And I think that's a very important thing to remember.
But at the same time, if they don't go out and vote, then they're also at fault.
They are at fault, but they didn't specifically choose this. So don't go with the assumption that there's some kind of mandate for Trump to be doing all this stuff, because there isn't, because a huge majority of Americans did not sign up for this.
How do you get people to vote, Alan?
Well, that's tough. I'm kind of hoping that more will show up to the next election because they'll notice that maybe politics actually does matter to their lives. When they find that, all the programs that they depended on and didn't realize have now been canceled.
It's a shame because voting is a privilege and these people are just throwing it away.
Yeah.
I noticed that the percentage of people who didn't vote is decreasing over the years, right? It was 48% in Reagan. Now we're at 36.
Yeah, there was a drop after the 2000 election. People suddenly realized maybe their vote could actually count, I think.
Yeah.
And then it's, kind of gone down from then. But that's the real key.
The thing is the Electoral College really warps the mandate thing, right?
But if that 36% showed up, there wouldn't even be a discussion about the Electoral College. It would just be,
It's true.
Yeah. Show up, folks. Yeah. Come on. That's great. I like that very much. All right. My pick is a video on YouTube called How One Company Secretly Poisoned the Planet. Now, this is all about Teflon and those other compounds that, what the hell are they called?
Forever chemicals?
The forever, but I want to give the correct
PFAS?
name. PFAS. Polytetrafluoroethylene.
PFAS,
or polyfluoroalkyl substances, PFAS, okay? Yep. So this video is cool because it gives the history of Teflon. DuPont was trying to figure out how to make refrigerators safer because they were blowing up because of the refrigerant. So they came up with this stuff called, which wasn't called Teflon yet, but it's basically a string of carbons with fluorines attached to them. And it turned out it polymerized into long chains and made this powder that couldn't be dissolved in anything. And during the Manhattan Project, they actually lined all the pipes with this stuff because there were very corrosive chemicals being used, and this preserved that. But nobody could figure out how to make it soluble so they could spray it on surfaces like a frying pan, right?
Hmm.
So they added these PFAS chemicals, which you can see in the video how they allow this long chain chemical to be soluble. And then that enabled them to spray it onto frying pans and all kinds of other surfaces. Even dental floss has this stuff on it. But the problem isn't Teflon. The problem is the PFASs that they use to get Teflon soluble. These are frickin' everywhere now. They don't degrade. They're and every time it rains PFAS. You know, the EPA has established limits in drinking water and in human tissues because it's in us as well. It's in polar bears, in the Arctic. It's everywhere. I don't know how it got everywhere. But, you know, a lot of the plants, DuPont in particular, spewed out this stuff in their waste streams, and it got into the environment. So this is a great video with some chemist who knows what he's talking about and talking about, you know, what are the consequences here and what can we do? And the thing is, you know, the EPA has set standards, but who knows if the EPA is going to be around? And, you know, the administration doesn't want people to know about this. They don't want to worry about PFAS because it will cost industry money.
I mean, I think a lot of people are starting to talk more about these PFAS chemicals. And in certain regions, like in the European Union, they're trying to phase them out, actually. And they're going to soon not be producing them anymore, which I think is very good. But right now, it's almost impossible to not be exposed because, like you said, it's everywhere.
So funny, when they first discovered that there's a guy in West Virginia who owned a cow farm. His cows were all dying because they were drinking the water that was coming out of the DuPont plant. And so that brought attention and they sued DuPont. So what they did is made a shorter version, gave it a different name so people thought, but it had all the same properties and all the same problems. And they will not admit to this day that they did anything wrong, you know.
Of course
Well,
not.
if they admit, then they're liable.
Yeah. Anyway, the science is pretty good in explaining this, right? So it is science for lay people. I really liked the explanation. They have pretty good graphics. It's some parts are over the top, but I liked it a lot. So, you know, there are tons of videos about this, but I watched just this one. It's pretty cool. All right, that's TWiV 1221. You can find the show notes at microbe.tv/TWiV. You can send your questions, your comments, your picks of the week to TWiV at microbe.tv. And if you enjoy these programs, we'd love to have your support to continue them. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Angela Mingarelli is at McGill University, Immune Vet on Twitter and Blue Sky. Thanks, Angela.
Thank you. It was a great time.
Good to see you again.
See you soon in person.
Yes.
It's coming up.
I haven't seen you in a long time, right? The BAT meeting was probably the last time.
No, you came to Montreal. You came to give a talk, remember? Last year.
2023, right? Or 24?
Wasn't it last year?
No, I don't think so. I think it was 23.
Really?
Yeah.
And then I went to New York. Was it after that?
You were in New York for TWiV 1000.
Exactly.
But that was April 23, I guess, right?
And then I think you came to McGill, though. I think it was 2024, wasn't it? Early?
It would have to be early then, 2024, yeah.
Yeah, to our seminar series.
Yep, I'll be up there in July.
Mm-hmm.
I'm Vincent Racaniello. You can find me at microbe.tv or on Blue Sky, microbetv.bsky.social. I'd like to thank the American Society for Microbiology and the American Society for Virology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.